Yes, we can hear you.
Okay, thank you. Okay, well, thank you very much, Reza. I'm very pleased to have the opportunity to speak about some of our research over the last few years. And I'll be talking in particular about life history correlates of human ejaculate quality. Now, life history theory addresses organisms' resource allocation decisions for competing tasks over the lifespan, and three, among many, key life history trade offs to which humans must allocate resources include the following, and I'll just list them here and then I'll speak about each one in turn. These trade offs include present versus future reproduction, quantity versus quality of reproduction, and mating versus parenting effort. Now, first, present versus future reproduction entails allocating resources to early reproduction at the cost of growth and maintenance, or growth and maintenance at a cost of delaying reproduction. Quantity versus quality of offspring entails allocating resources to producing greater number of offspring, increasing the chances that one or more will survive to reproductive age, but at a cost of decreased investment per offspring, or producing higher quality offspring by investing more in each, but there again at a cost, and that is a cost of producing fewer offspring. Finally, mating versus parenting effort entails allocating resources to high mating effort to increase offspring number, or high parenting effort to increase offspring quality. Now, these resource allocation strategies require trade-offs because individuals have limited resources to allocate to such tasks. These resource allocation decisions are manifested as phenotypic traits, including physiological, behavioral, and psychological traits. This phenotypic variation, therefore, can be thought of as reflecting resource allocation strategies, or what are called life history strategies, by which individuals attempt to optimize their resource expenditure. Now, human life history strategies and other organisms' life history strategies can be conceptualized on a slow-fast continuum, whereby life history strategies reflect coordinated patterns of traits, coordinated patterns of phenotypic traits. Relatively fast life history strategies entail greater allocation of resources to mating effort to increasing offspring number, whereas relatively slower life history strategies entail greater allocation of resources to parenting effort to increase offspring quality. Now, research applying life history theory to humans focuses on identifying physiological, psychological, and behavioral traits that comprise these relatively fast or slow strategies. So, for example, research has mapped personality traits, both normative and pathological, onto this life history strategy continuum. Other traits have also been identified to map onto life history strategies, including a co-vitality component representing health, and physiological correlates, including androgens, testosterone, and estradiol. Now, mapping individual differences on the slow-fast continuum of life history strategies has improved our understanding of adaptive phenotypic variation within humans. Okay, I mean, there are lots of questions that remain, but I think there have been some important advances. What I want to do today is I want to talk about ejaculate quality and whether or not ejaculate quality may also be associated with phenotypic indicators of life history strategies. Now, human ejaculate quality has in fact been shown to correlate with men's trait attractiveness. Trait attractiveness, in turn, is correlated with relatively fast life history strategy indicators in humans, including greater offspring number and greater number of sexual partners in both traditional and contemporary samples. These associations accord with what is known as the phenotype-linked fertility hypothesis. Okay, and this hypothesis, originally presented by Ben Sheldon, predicts a positive association between ejaculate quality and sex-typical male traits that confer reproductive benefits to men in short-term mating contexts. Now, situated within a life history framework, the phenotype-linked fertility hypothesis suggests that men pursuing a relatively fast life history strategy, that is, allocating greater resources to mating effort, will produce higher quality ejaculates to facilitate fertilization in short-term mating contexts. Alternatively, an argument can be made that men pursuing a relatively slow life history strategy may in fact produce higher quality ejaculates. Because human males invest substantially in putative offspring relative to other primate males, cuckoldry, which refers to unexpected non-paternity, would have inflicted reproductive costs on paternally investing men over human evolutionary history. Ancestral men may have guarded against cuckoldry by allocating greater resources to a single partner and their offspring or putative offspring together. These are indicators of a slower life history strategy. So this greater resource allocation to parenting effort versus mating effort, therefore, may be associated with higher quality ejaculates to ensure investment in genetic offspring. This cuckoldry risk hypothesis proposed here suggests that men pursuing a relatively slow life history strategy, that is, allocating greater resources to parenting effort, will produce higher quality ejaculates. So, in summary, what I'm going to talk about today is I'm going to share with you some recent research that offers initial tests of these two competing hypotheses. Okay, the first, the phenotype-linked fertility hypothesis, predicts that men pursuing a relatively fast life history strategy will produce higher quality ejaculates to facilitate fertilization in short-term mating contexts. And the cuckoldry risk hypothesis, which predicts that men pursuing a relatively slow life history strategy will produce higher quality ejaculates to reduce cuckoldry risk in long-term mating contexts. And what we did is we analyzed data from a larger experimental study that secured several self-report measures, including a widely used measure of human life history strategy. And we obtained 2 ejaculates from each participant. Using these data, then, we tested whether life history strategy indicators are associated with ejaculate quality across several clinical ejaculate parameters. So in terms of how we did what we did, we had our participants, which were recruited via ads posted on the campus of a university in the Midwestern United States and in surrounding communities. Now, men were eligible to participate if they met several criteria. They had not had a vasectomy. They had never sought treatment for infertility. They were aged 18 to 35 years, and they were currently in a committed sexual relationship lasting at least 3 months with a woman who also was aged between 18 and 35 years. And these age-related eligibility requirements were implemented due to the known declines in fertility for both sexes, though, of course, the fertility decline is less dramatic for men than for women. And potential participants contacted the lab to schedule 3 in-person sessions, okay, an intake session followed by 2 sessions at which they delivered a masturbatory ejaculate, about which I'll say more. The original data set included responses from 66 men with ages ranging 18 to 34 years. Their mean age was about 23 years. Mostly, about 91% of them were not married, but they were in relationships ranging from 6 months to 123 months. The average relationship duration was about 33 months, so just under 3 years. Now, the target sample size when we conceptualized this study was 100 men, and this was determined based on the primary aim of our study, which was a within-subjects experimental design. I will say that after 3 years of data collection and prior to data analysis, a consensus among the researchers was reached to stop data collection. We sort of needed to get on with it. Now, what I'm going to do today is I'm going to present only data from men who provided ejaculate samples for both masturbatory sessions and had complete intake session data. That turns out to be 41 men out of the original 66 included in the analysis. And I'll note that 14 of the men did not submit, 14 of the men who were not included did not submit 2 masturbatory samples, and 11 men who were not included did not provide complete intake data. Of those 40, given those 41 men, their ages ranged from 18 to 33 years, mean age of about 23 years, and the relationship length ranged 6 to 123 months with an average of about 36 months. So about 3 years. So all the procedures were reviewed and approved by the Institutional Review Board of my home institution, Oakland University. And men arrived to the lab at a scheduled time and date for an intake session. They were escorted to a private computer station by one of the researchers. Uh, Now, men were provided with a consent form and shown a video that described the various procedures that were involved. Consenting participants completed an intake session that included several self-report surveys on a computer, including the Mini-K, and that is an assessment of life history strategy along this slow-fast continuum. And the Mini-K includes items such as I avoid taking risks, and I would rather have one than several sexual relationships at a time. And these items were rated on a 7-point scale from 1, disagree strongly, to 7, agree strongly. And composite scores, which were comprised of a mean average for the Mini-K items, which had an alpha of 0.81. These composite scores were calculated, and higher scores on the Mini-K indicate a relatively slower life history strategy. Now, prior to each masturbatory session, participants received materials to collect and transport their ejaculates, including a non-latex, non-spermicidal condom, a plastic twist tie, a screw-top specimen container, a biohazard Ziploc bag, aluminum foil, and paper bag. Now, following World Health Organization guidelines, participants were instructed to abstain from ejaculation for 48 hours, but not longer than 7 days prior to each scheduled masturbatory session. These participants were able, in fact, to reschedule subsequent sessions or to request replacement materials, for example, if a condom broke, without penalty. And these masturbatory sessions followed a within subjects design whereby participants received written stimuli describing one of 2 scenarios. Of course, they each completed each one of the scenarios at different points. In the first scenario, this was one in which the participant discovered a partner's infidelity and then provided the ejaculate, or a scenario in which the participant discovered that their partner lost notable sum of the couple's money gambling. And I'll note here, and this was reported in previous research, that no difference was detected for any of the 17 semen parameters across the 2 conditions. And I'll say a bit more about that in just a moment. The order of the scenarios was counterbalanced across participants. Men were instructed to think about a scenario while masturbating, to not use any materials we did not provide. So, for example, to not use lubricant, and to masturbate without the help of their partners. Participants masturbated to ejaculation in a private location of their choosing while thinking about the provided scenario and wearing the provided condom. Participants then transported the ejaculate to the lab using the provided materials within 1 hour of ejaculation. Okay, well, upon delivery of the ejaculate, participants recorded on a self-report survey the time since their previous ejaculation, in addition to the time of their current ejaculation, to confirm that they had followed the instructed procedures. All participants produced and delivered ejaculates in self-reported accordance with the instructions. That is, for example, that all the ejaculates were packaged properly and so on. Ejaculate quality was then assessed using the Semen Quality Analyzer, or SQAV. This is a machine that's produced and sold by Medical Electronic Systems out of Israel. It's a fully automated machine that analyzes semen along 17 clinical parameters using electro-optical technology, signal conversion, and the application of a variety of proprietary algorithms. Upon receipt of the participant's masturbatory ejaculate at sessions 2 and 3, the entire ejaculate was syringed into a proprietary measurement capillary, which was then inserted into a chamber in the SQAV for automatic analysis. After completion of the automated analysis, all materials that directly contacted the ejaculate were then discarded in a biohazard waste container. And I'll note that the time between each masturbatory session ranged from 2 days to 28 days, and on average was about 7 days, or a little bit less. All men were compensated $25 at the completion of each session. What did we find? Well, let me note first that the SQAV estimates 17 continuous clinical semen parameters and it does this in about 75 seconds. So it's quite remarkable technology. Higher values in each case reflect higher quality. Okay. What we did is we calculated the mean average of each parameter for each participant across the two samples, the two masturbatory samples, to increase the reliability and power of these statistical analyses, particularly given the relatively small sample size available for analysis, which was 41 men. We computed zero-order correlations and partial correlation coefficients, controlling for participant age and abstinence time prior to ejaculation, between scores on the mini-K and each of the 17 clinical parameters. Now, the sample size, such as it was, did in fact afford adequate statistical power, about 80%, to detect effects of moderate magnitude, around 0.4 in terms of the correlation coefficient. And I want to note that 10 of the 17 zero-order correlations and 6 of the 17 partial correlations reached statistical significance at p set to less than 0.05. And importantly, these correlations were uniformly positive, okay, indicating that men pursuing a relatively slow life history strategy produced higher quality ejaculates. Now, let me just highlight a few of these results. I'm sorry for the flurry of data here. This is showing you the correlations between each of the clinical parameters, semen parameters, and bivariate correlation with the scores on the mini-K, and partial correlations with scores on the mini-K, controlling for the man's age and the abstinence duration. And I'm going to highlight 3 parameters. The first is the concentration of progressive sperm that are shaped normally. You can see the correlation with mini-K scores is 0.42. The partial correlation, controlling for age and abstinence, is 0.35. Okay, I'll next highlight the percentage of rapid progressive motile sperm. The bivariate correlation is 0.45. The partial correlation is 0.41. And finally, I will note the sperm motility index, and I'll say more about this in just a moment. You can see the bivariate correlation with scores on the mini-K is 0.57, and the partial correlation, controlling for age and abstinence duration, is 0.54. So correlations between scores on the mini-K and the sperm motility index, and this is an index of fertilization potential based on sperm motility and velocity, showed the largest associations. Again, the zero-order correlation was 0.57, the partial correlation was 0.54. And just for sort of graphical presentation purposes, here I'm showing the zero-order correlation between the sperm motility index and the mini-K scores. You can see that it is a positive relationship there. The R-squared linear is 0.323. So what these results provide is, at least preliminary support for what we've called the cuckoldry risk hypothesis. Men that reported relatively slow life history strategy also produced higher quality ejaculates. So let me remind you, the current study tested 2 competing hypotheses, regarding associations between life history strategy and ejaculate quality in humans. These were the phenotype-linked fertility hypothesis, which predicts that men pursuing a relatively fast life history strategy will produce higher quality ejaculates, and what we've called the cuckoldry risk hypothesis, which predicts that men pursuing a relatively slow life history strategy will produce higher quality ejaculates. The results, such as they are, do accord better with the cuckoldry risk hypothesis, insofar as men pursuing a relatively slow life history strategy produced higher quality ejaculates. Now, the sperm motility index, that is an overall, quote, quality score based on sperm motility and velocity, and that did show it showed the large association with scores on mini-K, on the mini-K assessment. And these were robust to effects of age and time since last ejaculation. And importantly, among those parameters that are assessed by clinicians, sperm motility is one of the most reliably associated with conception in humans, suggesting that paternally investing men produce ejaculates with higher fertilization potential than men pursuing a relatively fast life history strategy. Now, the empirical application of life history theory to humans includes attempts to identify coordinated patterns of phenotypic traits that comprise relatively slow or fast life history strategies. In the current research, we investigated whether ejaculate quality may be a phenotypic trait associated with life history strategy in humans. These results suggest that ejaculate quality does reflect resource allocation decisions for greater parenting effort as opposed to greater mating effort. Now, that men pursuing a relatively slow life history strategy produce higher quality ejaculates accords with research documenting positive associations between human ejaculate quality and long-term, but not short-term, male attractiveness ratings, given that the traits associated with long-term partner attractiveness reflect a relatively slower life history strategy. Alternatively, the association between relatively slow life history strategy and higher quality ejaculates in our sample may be due to the relationship that each variable has with a third variable, namely general health. There is a robust association between human ejaculate quality and indexes of male morbidity and mortality risk. In fact, within the human life history literature, lower morbidity and mortality risk are key factors assumed to be associated with relatively slower life history strategies. So given that morbidity and mortality risk are associated with human ejaculate quality and life history strategy in humans, and we're suggesting that future research should explore this potential alternative explanation for the results that we report here. Now, that men pursuing a relatively fast life history strategy produced lower quality ejaculates may be attributable to potential long-term trade-offs over the lifespan, such as a developmental trade-off between investment in secondary sexual characteristics and investment in ejaculate quality, or downregulation of ejaculate quality to afford production of more frequent ejaculates, or to potential short-term trade-offs, such as diverting sperm production resources to courtship behaviors, or downregulating sperm production in non-competitive short-term matings. Now, all of this is to say that each of these non-mutually exclusive explanations has been suggested for parallel findings in non-humans. The developmental mechanisms underlying potential long-term and short-term trade-offs for ejaculate production in humans, however, remains a valuable area for future exploration, and for which there's not a good deal of work. Now, research on human ejaculate quality certainly has focused on testing the phenotype-linked fertility hypothesis on the assumption that ancestral men pursuing short-term mating would have produced higher quality ejaculates more capable of fertilizing ova. Guided by a life history perspective, however, the current results suggest that ejaculate equality accords more strongly with parenting efforts than with mating efforts. Human males invest substantially in putative offspring relative to other primate males, and therefore, cuckoldry would have inflicted significant reproductive costs on paternally investing men over evolutionary history. Men pursuing a slower life history strategy may prioritize investment into higher quality ejaculates rather than, for example, secondary sexual characteristics to mitigate cuckoldry risk. Okay, that's the argument anyways, thereby increasing the likelihood that resources are invested in genetically related offspring. Now, there are a slew of limitations to the research that I've just presented to you, and let me just highlight a couple of them. Certainly, additional research is needed to identify which phenotypic traits of men are reliably associated with ejaculate quality, and to confirm whether ejaculate quality is reliably associated with other measures and indicators of life history strategies. The measurement of within human variation of life history strategy has been the subject of a good deal of debate since the data reported here were collected. And we've suggested, and we do suggest, that future research might employ multiple measures of life history strategy to afford assessment of the validity of the life history continuum construct. Characteristics of our sample should be considered when interpreting the results. The sample included men who were self-reportedly in committed relationships and therefore may be biased toward assessments of men pursuing a relatively slow life history strategy. So clearly, future research could collect multiple ejaculates from men with greater variation in life history strategy. We note, of course, that our sample resided in a suburban Midwestern United States sample. I mean, it was a rather circumscribed sample. We don't know, but the results may or may not be generalizable to different populations within the U.S. or, for that matter, across different cultures. The sample size was small, and it was limited to the detection of moderate to large effects. Smaller associations of ejaculate quality with life history strategy certainly may have been missed. Our small sample also increases the possibility that correlations reported here are unstable, meaning that effects, they may be variable in subsequent replications. We just don't know at this point. Now, several procedural aspects of the current research also should be considered when interpreting these results. Due to ethical and biosafety controls implemented at the university level, we simply had to rely on participants' confirmation that collection and transport protocols were followed. And that may have been a big ask. And although participants confirmed that they followed the protocols, we cannot claim the degree of certainty afforded if the collection of, if collection had occurred in a controlled laboratory environment. Okay, let me offer a couple of concluding comments. Identifying patterns of phenotypic traits associated with relatively fast or slow life history strategies may provide insights into resource allocation decisions. The current study investigated associations between life history indicators and human ejaculate quality to test competing predictions derived from the phenotype-linked fertility hypothesis and the cuckoldry risk hypothesis. Now, the results, such as they were, accorded more strongly with the cuckoldry risk hypothesis. In short, men pursuing a relatively slow life history strategy produced higher quality ejaculates, perhaps to reduce the likelihood of cuckoldry. Although broad patterns of phenotypic traits have been associated with mating and parenting outcomes, much less is known about physiological and reproductive mechanisms underlying life history strategy in humans. The current findings at a minimum contribute informative data on on the relationships between physiological and phenotypic indicators of life history strategies. Let me close by acknowledging the time, effort, and investment of my doctoral students at the time, Nicole Barbero, Andrew Holub, Austin Jeffrey, and Guillermo Lopez, and my postdoctoral student and lab manager, Tara DeLise, and my faculty collaborators, Lisa Welling and Virgil Ziegler-Hill. And finally, please let me note the funding, which was provided by Oakland University's Office of the President, Provost, the Dean of Arts and Sciences, and the Center for Biomedical Research. Thank you so much for your time and attention. I appreciate